So, good morning, Namaskar, Khurumjuri. So, respected Director Sir.
Another word, what you use?
Khurumjuri means Namaskar in Manipuri.
Kurumjiri. Okay. In which language it is?
Manipuri.
Very good. Yeah.
So, respected Director Sir, and today guest, Head Plant Protection CTCRI Trivandrum, and our in charge of the three departments, as well as principal scientists, senior scientists, and technical staff. So today my topic is bottom-up effect of on the plant insect interaction in horticulture crops. Because I choose this topic is related to host plant resistance, and that is the first component of our IP. So when I joined CIS Bikaner as a scientist in 2009, I seen one train that came from Punjab to Bikaner. The train name is cancer train. So I thought, what is the major problem in this area? So that most of the people told that maximum pesticide we are using in Punjab area. That's why each family having one cancer patient in each family. So that's why that all are coming at Bikaner because government hospital is very good. So the train name is cancer train. So I thought we are doing against the chemical insecticide. So that's why we choose this host plant resistance, because in my institute around 15 to 20 crop is there, and each crop having more than in bear having more than 300 germplasm, like other kasri having 65 germplasm, pomegranate 165 germplasm, that is there. And no scientist is in entomology is working before my, so I start my career from this, and that is also basic component of natural farming and our integrated system. And other also it's a relatively economical after establishment. This is also very important, no environmental effect, and it's good for the development of resistant varieties, and as well as market development in plant breeding program we can also use as a resistant genotype. So this IPM system is the first that is point. This is host plant resistance, and it is also known as bottom effect because it's coming from the seed or it's coming from the plant. That's why we give the name is bottom effect. If biological control we are using, that we told top down effect, because after insect is coming, then apply this biological control. That's why that name is coming. And why using this name bottom effect? Because I submitted 2-3 times that simply host plant resistance study in this crop, but many journals rejected that paper. After that, we are putting that name bottom effect, and it's selected in crop protection. So I thought it's very good the title. So what is the interaction? This is also very important because that plant and insect both are. Yes.
Previous slides.
Yes, sir.
One thing you have not taken.
Actually, sir, this is I take only this host plant resistance. That's why we are not explained.
Okay, one of the components is that.
Yeah, only one component, and that is the first component of IPM. That's why also important. Other also regulatory measures, cultural measures, biological control also there, botanical pesticide also we taken one patent also on this. Then last component is chemical control. But in India, 90% we are all the farmers using the first component as a chemical. So this is we require this type of system. And if you are coming on interaction, that insect and plant that existed in for millions years ago, and it's sometime offensive, sometime defensive work is doing both plant and that insect. And what type of mechanism is found in plants? That is there. First is antigenosis, means some specific character is found on the plant, like morphometric characters, like some trichomes is there, hairiness is more there if you have any line or genotype or that is there. So that also resistant to any insect pest or diseases. So this is also very important mechanism. Then second is antibiosis, means some allochemicals like phenol, flavonoid, alkaloid, tannins. That is, if you are more is there in plants or genotype that having resistance, that is resistant to insect pest. This is also we found. And last is tolerance. That may be coming due to this maybe due to antibiosis, antigenosis, or tolerance. That is withstand after damaging, also withstand no any effect is plant. So this is part of this. So yes, sir.
No effect on the plant.
Plant is damaged, but we just again maintain that our body or the plant of this. This is there, and tolerance. Tolerance is the part of resistance, sir. In mechanism, that tolerance also there, antibiosis is there, and then antigenosis is there.
The definition is different.
Yeah.
Definition of tolerance, you should know.
Okay.
Because in so many times this is wrongly used, the tolerance. Though the damage is there, the plant can able to give normal yield.
Normally.
Normal yield.
Yes.
That is the definition for the tolerance.
Yes, sir. Yes.
Okay.
Okay. So on the basis of this, we are taking first is Bear. Bear is the major area in my institute, Central Institute for Arid Horticulture, that I completed 10 years in this institute. So in this, that bear fruit fly, Carpomia vacuna, is the major pest, and it's a monophagous, because in most of the cucurbits that all are polyphagous, like Bactocera cucurbiti, Bactocera dorsalis, Zonata, that many is there. But only this one species is there, Carpomia vacuna, that only attack on the bear and this family. So that's why we are taking, and around 60 to 70%, if you are not controlling, then 60 to 70% that damaging to the plant. This is also there. And on the basis of, we have taken 3-year study. First, we taken the 319 germplasm as a preliminary study, and then we found some are susceptible, some are highly susceptible, some are resistant, some are highly resistant, moderately resistant. On the basis of this, we taken around 30 to 35 genotype or germplasm for final study like antibiosis and antigenosis study. Then on the basis of this, we found that Elaichi, that having only 15% damage, and it's come under resistance category. And another is Tikadi and Kata. 3 lines we have identified that is resistant to fruit fly. And these are the different type of fruit or variety or that lines we are taken in final stage. And this is we also correlate. And we found that if you are phenolic content, tannin content, flavonoid content, that are negatively correlated with the fruit infestation. So this is also very good to be found. And if you are fruit width, that is not important, and fruit length also there. But pericarp thickness, this is also some relation is there, negative correlation we found. So on the basis of backward stepwise regression, we found which molecule or which that antibiosis character that is more role in the resistance. So like flavonoid content is the maximum role for on the basis of this backward stepwise regression, that around 83%. Maybe some another also there, but on the basis of this analysis we found. Another we have also found that phenolic content around 5.5%, that major role for this controlling this fruit fly. And on the basis of principal component analysis that we are making, these are the resistance line, or these are the moderate resistance line, and these are the susceptible line, and these are the highly susceptible lines. And then this we have published as a paper on crop protection, as well as we also received the yeah.
But all those accessions are partially correlated with your sense of chemicals.
No, all are not found, sir. Some are less, some are high.
How it is proved?
On the basis of this, sir, backward stepwise regression, as well as on the basis of correlation.
No, that is on the basis of infestation, resistance it showed. No?
Yeah.
Resistance it showed, your group resistance, susceptible, moderately, all these things.
Yeah.
But that is positively correlated with your antibiosis and other things. Did you correlate with the hairiness?
Antibiosis as well as sir antigenosis, both we are taking.
But that table is there.
Yes, there, sir.
Okay.
Complete. If you are seeing that.
No. Are you going to project that the all these resistant lines contains more of phenolics?
In this paper that is there, sir. But we are not presenting because we are presenting many is there. That's why not presenting.
Okay.
But in this paper, that all complete phenolic content and all which line,
And
how many percent.
resistant plants are having more of a phenolics compared to
Yeah.
the sustainable one.
Yeah.
Okay.
If your resistance is there, then phenolic compound is more is there.
Okay.
That's why negative correlation is there.
Okay.
And pericarp thickness also major role for controlling this. If you, and hardiness also, we are taking the tensiometer is there one. So we are taking hardiness of the fruit also.
This has been same parameters have been found in other crops as well.
Other also we are taking. Actually in cucurbit, sir, we found that length of ovary pubescence. Ovary is the main, that female ovary is there. I will discuss in next.
It's a different character.
Yeah.
What you are telling, the pericarp thickness.
Yeah.
It is the same thing is applicable to other crops, or it will be
Other maybe you can take, sir. That depend upon the which part is damaging. We are taking fruit fly is damaging to fruit.
fruit fly.
So fruit fly.
It is for fruit fly.
Only for fruit fly.
So the fruit fly is there in several crops.
Yeah.
So all those crops.
That we can. I also take in sir cucurbits also. That paper also published.
Guava, mango, banana.
That we can do, sir. Also other also there. And this already there, sir. We also deposited in technology data repository of ISHAR also. And we also publish one inventory of CIS technology for arid and semi-arid horticulture. And that this technology also published from this institute also. And another we are taking this, sir, kachri. That is also cucurbits, and it's only found in Rajasthan area. It's a undomesticated type vegetable is there, cucurbits. So only in Rajasthan is found cucurbits, sir.
This is
Cucumis melo. Cucurbits, sir. Cucurbitaceae family, and Cucumis melo variety calosis is there, and near to our muskmelon. That is variety is different, but species is the same. It's only found in Rajasthan, sir. And in our institute, one variety released. So if you are when selling that seed, that police is coming because it's very high demand of these lines AHK 119. So yeah.
Phenols and other things. Phenolics, flavonoids, or some volatiles, something like that. Or screening, whether we can use something, some sensor like we can develop this. Then
I have own sensors on how to some electronics engineering can develop them.
but collaboration with the engineers.
If we find a good this thing, I don't have any expertise in sensors.
So there is Dr. Ravi Narayan South in IARI who's working specifically on these sensors, and he's working on such type of sensors that you want. Chemical based.
Of sensor weight is person, and then the validation by yes, just what I am telling is just you have some idea on that.
But if we get the sensor, then we can develop some ICT based system through which it can be validated.
Yeah, that's what some collaboration.
Sir is developing the sensor, sir. Based on it.
Just you contact with those persons, you take home message and tell him something. Any presentation, how to take it forward, or what idea we can take it, so that from that we read what we are getting.
But has he in public domain released some sensors?
No, he is working. Actually, sir, there is one sensor. There is one sensor which is even detecting the fruit so on that basis उसमें भी वो है ना कि जो भी chemical changes हो रहे हैं उसके basis पे they will come to know shelf life मतलब उसको
Note it down and we visit one day to National Physical Laboratory and what we are getting.
Yes, sir.
So that this type of things, just download some papers out there.
Yes, sir.
Also download some papers.
Okay, sir.
The volatiles and other things, the phenolic and some flavonoids, something like that. No? Suppose taking the extract, put it in sensor so that how much it is there. Okay. So the genotypes, whatever they are getting, we can produce and then we can bring to the crop-based input to screen the things for resistance.
Yes, sir. Yes, sir.
So that type of things. This is a new idea. That's what we expect now.
Okay.
We because the time consumption is more because of regular.
specifically.
And also it will be more accurate. So some new technology we can develop. Okay. So yeah, proceed.
Okay. Actually, sir, in botanical pesticide we are restricted now, sir. That also same condition is there. If you are more flavonoid, phenolic, and tannin content is there, that is very good pesticide. This is also there. Some are repellency, some are deterrency. This is found in the that by pesticide.
You can also be more. And the area is also, we will discuss a lot of this type of things.
So we are taking, sir, second crop is Kasri, and in my institute that having 68 germplasm. So first we screen preliminary screening, same like susceptible, highly susceptible, resistance, and high resistance. Then on the basis of incidence, we are taking the larval density as well as percent infestation. After that, we around 20 to 25 germplasm we are taking for biochemical as well as antigenosis or antibiosis characters. So on the basis of this, we found that that IC number that 350933 that having only 5 to 5.7 that incidence is there, and it's highly resistant because if you are less than 10% incidence is there, that come under highly resistance lines is there. Then another also that is resistance lines, and some are highly susceptible lines also we found, and on the basis of this that we take the principal component analysis, and then we found that this line is on the basis of both characters, sir. We are taking antibiosis character also, then we are taking antigenosis character also, morphometric as well as this, and we found that this variety is 119 as a resistant to fruit fly, and then already released from Center Variety Release Committee also and from Institute also, sir. And these are the different germplasm is there. And we have taken all the IC number of 68 germplasm.
This is a hair and trichome and
Yeah. So I will see, sir. This is the correlation. We found that flavonoid content, PC content, and tannin content, and total alkaloid content also. So all are that negative correlation is found. And on the basis of this, we found that flavonoid content is major role, but followed by tannin also major role around 2.4%. Then other also 0.4% or other. That though on the basis of backward stepwise regression, and this is related to antigenosis characters. So we found, sir, length of ovary pubescence. This is major role for controlling this fruit fly, and you see, sir, that is the length of ovary pubescence 84%. In overall contribution of we are taking 4-5 that characters in which length of ovary pubescence is made.
So the other characters.
Other character like rind hardiness also.
No, previous slide.
Yeah.
Yeah, one, then?
One is length of ovary pubescence we are taking. Another is rind hardiness, means fruit hardiness. Then rind thickness, how many thickness is there in that Kalsari crop? Outsider. Then fruit length and fruit diameter, that is no.
Okay, one is rind thickness and rind hairiness.
Yeah.
And ovary, what is that?
Length of ovary pubescence. This is there, sir. This I will see you, sir. This ovary. Actually, in sir cooker beads, male and female is different. Male having only flower, but female having the ovary. If we are it's pollinated, then big size fruit is coming. But initially, if you are not pollinated, then same this side and drop it. So this is the specific character for cucurbitaceae family. And you see, this is the hairiness, sir. So this is because in initial stage that egg laying done by this fruit fly. So
instead of that, 1 minute, instead of putting so much, take one resistant plant, one moderate resistant, one susceptible.
Yes, sir.
The hairiness you project it, focus it, so that in some analysis or something like that, you focus this one, the hair, how much this one. This is more important.
Yeah, sir. That in table that is there.
No, table okay. I'm telling when you are projecting it. Don't project this much.
Okay.
Only 3: resistant, moderation, susceptible. So that gradually it is decreasing or increasing, whatever it is, depending upon the character.
Okay, sir.
So if you are projecting in a large size, visible this one, then it would be very nice.
Okay, sir.
Some analysis also you can do. Some it is difficult, I think. By which this one you can focus it, that you can do it. Some analysis you do it.
Okay.
For the at least for any success stories or something like that, if you want to, okay.
Then on the basis of this, we have already published this paper in crop production. Also, we release the variety because we are going not for publication, sir. We are also developing the product also. And already that ISA technology data repository we are also submitted and also published separate.
Which product you develop?
Variety, sir. We have developed na variety.
Variety. This was done some other institute, right?
No, my institute, sir.
Okay, so that is some other institute.
No. CIH. Central Institute for Arid Horticulture. I am worked for 10 years, sir.
You are sitting here.
Sir, that my work is
Okay. That's what I am telling. It was done in some other institute, right?
Yeah. I completed 10 years in this institute.
Okay.
And we develop this variety.
Now you have not come out of your institute.
That 10 years, sir. We are here.
10 years. That is different. Okay.
Okay, sir.
Yeah.
So next is we are taking a ridge guard and we have 35 germplasm of this and same we are taking in this also highly susceptible.
Okay.
And on the basis of this we are found that AHRG29 line that is resistance.
Okay.
And this variety also already has a Thar coming we released from institute as well as Central Variety Release Committee. And this is the relation same that we are taking in Kasri.
Okay.
Yes, sir.
Same.
Yeah, same.
Same. Go to next.
So this is another sir tinged bug. Actually, in Rajasthan, sir.
This is fruit fly?
No, it's bug. Hemipteran bug.
Okay.
So sir, one plant is there, Lasoda, cardia mixa is there. So in this, that is the maximum incidence of this tinged bug. So in my institute, old institute, that having 23 germplasm. So on the basis of that, we found one that genotype that is resistant to this tinged bug, and this registered as a tinged bug resistant line and already registered in NPPGR, and we have also received the INGR number 21069.
Okay.
So this is about this. Actually, in this sir, we found that leaves actually this attacking on the leaf of the plant. So if you are less hairiness, less incidence is there, or more incidence is there. And if your hairiness is more on plants, that leaves, that more, and also hardiness. Leaves hardiness also major role for controlling this. So this is
hardiness in the sense it is cuticle or something like that.
That leaves
hardness. Which parameters? The hardness means. It is some parameters will be there.
Thickness also there, sir. Thickness also.
Thickness of epidermis or something like that. Leaf lamina.
Complete we are taking, sir, complete. And on the basis of tensiometer, we are putting how many pressure is required for.
Okay. That hardness gives which one gives hardness to the leaf?
Which means?
No. Which one gives any? I don't know. Something which gives hardness, no?
Sir, that like
which is influencing.
Sir, like tannil, phenol, flavonoid, that all are making the hardiness of the plant, sir.
Cells.
Cells.
Okay.
And
no, you just find
it out.
It out if anything specific, no? Among different parameters, which is which parameter is influencing more on the hardiness. If that is there, then that can be alone selected for screening the varieties.
That's why, sir, I work on this induced resistance and silica we are using when completed 3-month training. So that also we discussed.
Okay. Yeah, a little bit go fast. Otherwise, in between 10 minutes, I will not be there.
So this already
I will be missing.
Okay, sir. Registration and snake melon also there. That also underutilized crops in Rajasthan.
And wait, in this type of things, how will you go for IPM for the protection of the crop from the farmer's point of view? So some are okay. So this insects and this is some wilt and other things. Okay, but what about the viral disease? Who is working on viral disease and anything? can be done on that. Otherwise, it will be incomplete. Your IPM
yeah.
cannot be working in that.
Yeah, yeah, sir.
So somebody has to take up this type of
That
things
also
on viral
requires-
disease,
Mhm.
particularly the plant extract, botanic extracts, or the pathologist also should be involved. Should be taking the measures for this viral disease,
Yes, sir.
because the farmers he will not differentiate us
Yeah, yeah, yeah.
that you are entomologist or you are fungal pathologist, you are bacteriologist, so I need not bother for virus. Farmers want solution for his problem. So for that, how we are prepared
Yes, sir.
to this type of problem, that we have to see.
Okay, sir.
There is my specialization is on a bug and not the white flies or fruit fly, and you cannot tell, no?
Yeah,
So
yeah.
collectively, IPM means everything. It
Sure.
includes everything. So now you can note down something that the pathologists should also work on the botanical extracts for the management of viral diseases.
Yes, sir.
Whatever the management practices are there, or some induced resistance like some chemicals which is involved in induced resistance, that also can be bion or something like that. Some biocontrol agents also can be, because it will delay the expression of viral symptoms,
Yes,
or the biocontrol agents or some chemicals which can control the vectors so that the incidence will be less. So in that angle, you just try when you are developing a IPM module, that has to take care of the viral disease also. Okay.
Okay, sir.
So when you are presenting, all these things should come.
Okay.
Okay.
So that in our Snake Melon also, sir, we have 65 germplasm in old institute, and then we select preliminary screening. Then same condition in other we are doing. That is also we are doing, and also release one variety that is AHS 82.
Okay.
And it's very
That is
famous.
not important much. Only the bottom of this one, the
Yeah.
characters involved, you only focus.
Yes, sir. Yes,
If
sir.
the same
So that already that this paper is published in breeding science in Japan, and technology also released from institute, and variety also from Central Variety Release Committee. And we also worked on that. All works are related to last 10 years I done. That is presented, sir. That also I work on this pomegranate, and in pomegranate, sir, mite is the major problem. So that also in institute that having 165 germplasm is there. So we take for preliminary screening, and then we take the final screening, and we found that Achik, Dana, Aga, and so in this we are taking variety, already released variety also we are taking, and germplasm we are collected from all over the India. So that also taking for this, and we also analyze or take the height, canopy size, stem diameter, and phytochemicals like total phenol, flavonoid, and tannin. And then we correlate with the incidence. And this is already, this also published in agriculture, that MDPI journal is there. So I also worked for 3 months in Thailand when in Central Agriculture University. So my work on induced resistance, because it's also related to resistance. So I take different concentration of calcium silicate, and this is also very important. Just 3 days or 4 days before, we also online meeting with Australia. So that we already done, sir, in Thailand, or we are taking this.
The sources, you enumerate the sources of silica.
Yeah, sir.
Not only calcium silicate, but also the other organic based one. This is the chemical based. Chemical based silica sources. The other right, either other organics like fly ash is there. Then some ash from the rice husk ash.
Yeah, sir.
Yeah. So you consult with the madam also, and you enumerate these things, and also include in the IPM.
Okay, sir.
You have to note down then and there all these things, whatever.
We also observation like enzymatic activity, different type of.
That you can do it. Normal thing.
Normal things. So after that, sir, we take that in control, highly incidence of this apid is there. If you see 2% and 4%, that is non-significant. 2% and 4% is not any significant. But That's why we are putting that 2% is good for management of this aphid in rice. And you see, we are taking the electron microscope this photo and clear cut that that incidence is there in 2% and 4% and control. That clear cut is seen that silica is there through electron microscope. Also analyze the which is the highest percent is there silica, that around 14%. This coming through only electron microscope, sir.
So 4% is effective.
2% is effective, sir. 2 and 4 no any significant difference.
Some incidence was there in 2%. 4% it was looking healthy. Like
Only limited, sir, but is non.
Go back. Go back. You see here. You see the
First is the control that having the aphid population.
Yeah, second is
Second is 2%. That also having limited and 4% also there.
4% it is free, na?
Not free, sir. Free to no.
But the pink color or red color, what is that color?
That so, sir, that mature leaves is there some. Also,
No, the grain.
Grain also.
Rice aphid. What is the symptom of rice aphid? Can you just
no, below side is there, sir. In this below side is there, sir.
Oh, not on the grains.
Yeah. This is there.
Not on the grains.
Not on the grains. Sir.
But why you have projected grains?
That's the part of this, no, sir.
No, you don't project like that.
Okay.
Only the symptoms should be there.
Yeah,
What is the, see, when you are taking the photograph, which is your object, that only you have to focus. Not this type of things. See, for the common person, how I am telling, after seeing that red color, this one, I thought that it is a
no, that the maturity is there.
Rice aphid, it attacks only the leaf.
Leaf as well as near to that.
Okay. That before the panicle, panicle. That what is that? The parts which is bearing the grains. If it is sucking, then the movement of the nutrients will not go, na? So the maturity will not be there.
Yes, sir.
Yeah. Okay.
So on the basis of this, sir, electron microscope, we found which percentage of silica is there inside.
2% you are telling that is good. What about 4%?
4% near to 2%.
Nearer.
No significant.
That type of things you tell.
That's
no
why,
significant effect.
No significant.
What about 3%?
3% we are not taken, sir. In this. We are taking 3.
No, 2%. How much incidence was there? How much percent of infestation was there?
The data is there, sir.
Data, but I am telling. So suppose 80% something reduction is there. Compared to control. So if 3% is giving 95%, that you should select 3% because there's a gap between 2% and 4%.
3% we can.
So if you say that completely free from, I would not have asked this question. But you told somewhat better like that. So 3% you should.
To note there, sir. Completely note there.
Not there. Then you should have gone up to 3% also. Then that is the lead. That type of things you have to follow, no?
Yes, sir.
Once if you are getting some lead, then how to further fine tune your technology.
Yes, sir.
That is important. Okay.
Then this is the different concentration of this silica in the plant. And that already we have submitted this article.
Okay. Next.
So these are the team.
That's all over?
No, sir. This is complete up to HPR. But I also present 5 to 10 minutes that last 15 years what type of technology and publication we have done.
But this seminar part of this
seminar
one is over?
Yes, sir. So in last 15 years I joined, sir, 2009. After that we developed some technology like patent also, some variety also.
Quickly go.
Quickly, sir. That one patent on biopesticide.
What product it is?
Tharjaic 41 AC. Actually, sir, in Rajasthan, that one plant is there, Tumba. That is the weed, sir.
Botanical extract.
Botanical extract. And we are combined with the cow.
This is for which insect?
We are testing whitefly and jacid and helicorpa also, sir.
For same thing we can apply here. In IPM we can include.
That's why, sir, we are taking.
Okay, good. You will be developing all these.
10 liter already we taken from the institute.
It is insect specific.
Insect specific.
Not crop specific.
No. Insect.
Suppose aphids means wherever aphids are there, if you spray, it will control.
Yeah, yeah.
Did you validate it?
Yeah, sir. Already in institute, already validated, sir.
No, again institute. In how many crops you have validated?
Sir, I developed this in 2009. After that, 2020, I transferred from this to.
No. All those
So,
things I
Yeah.
validated, not validated.
Validated,
If it is
sir.
validated, how many crops? I'm not asking all the
In my
crops.
horticulture crops are already validated, sir.
Okay. Now you you just present to others.
Yeah.
I will just in 10 minutes I will come.
Okay, sir.
Okay. So later on this part, whatever the part presented in my absence, I will ask you. No. Wait. This. Okay, you can see.
Okay, sir. So this already that paper also published on this 41 AC. ICI news also coming. Yeah. Another.
नहीं वो बोल रहे हैं कि वहाँ सुनेंगे
पानी पी ले
चाय पी लो बोलो चाय आ गई थी सर जब तक
what I can suggest is you can little bit modify your product.
Yeah.
You develop here something you add.
Yeah, sir.
You add
I will try, sir.
Some more potential, something like that.
Yeah.
So that we can have a stimulant or growth promotion.
Yeah, sir.
We can distribute in farm. Little bit this.
Yeah.
Keep it there. In addition to that, add one or more, so that it will not be the same of what you have patented.
Yes, sir. That will do, sir. Okay, sir. So that's sir already that release also from the Honorable Agriculture Minister in 2019. Kailash Chaudhary ji and Arjuna Megwal is also there, and Aisha also published inside. So another I applied one patent on sir pollen substitute. Actually during
Keep it louder. Your mic is on.
Yeah, on. So sir, during Central Agriculture University, I applied one patent on pollen substitute during that period of honey bee rearing, so that we required the artificial diet. So in Manipur area, one plant is there, rice bean, black rice bean is there, beans is there, and we add some honey and other, and then we make this formulation, and it's also running. We are given also farmer too also, and it's under at present we are not receiving a PR of this, maybe coming early as possible. So this is also I applied, and these are the variety I associated in this variety. Already 8 variety released, and 3 lines we registered in NPBGR as a resistance, as well as one monoecious line also there, and 2 lines we taken IC number that from Northeast CIU. So this also we do.
Registered by Plant Germplasm Registration Committee.
Yeah. So another sir, this is related to our, because I am also in CIU also as a farm in charge. So we transferring the wetland around 15 to 20 hectare land is there, no any use. So for that we developing the wetland to farming land. So around more than 2 crore we are expanding for this development of this area. And at present you see this is the situation. We are making sunken bed, raised bed, and then that fish pond. So and that already that also published, and we have received the one award also for this. We are making one video and submit to manage. So we received 10,000 money also, and this is the video, sir. That online also there on YouTube. It's 10-minute video is there.
उसका वॉल्यूम वहां से बढ़ाओ, आपकी वॉइस भी इसलिए नहीं आ रही।
Wetlands in Manipur cover nearly 6.8% of the total land area of Manipur. Important wetlands include Loktak, Phumland, Lamjau, Khoidum, Kharung, Ikop, and Waithau. Common natural vegetation found in wetlands are water chestnut,
Initially, sir, this is the situation of this
water
farm.
spinach, fish mint, smart weeds, clover, lotus, etc. In alignment with Honorable Prime Minister Narendra Modi's Atmanirbhar Bharat, Central Agricultural University's present Vice Chancellor, Dr. Anupam Mishra, envisioned the conversion of the 15-hectare wetland area of Lumfel Path, which is under CAU, into farmland for the welfare of the local people. The central farm of the university, located at Lumfel Path, is developed in approximately 15 hectares out of the previously low-lying marshy lands which were left unused.
Before 2020, sir, nothing is there in this farm.
The name Lumfel Path means name of a place that
You see, sir, that is sunken bed and raised bed.
means
So in
lake.
raised bed, we
The
are using
state
the vegetable as well as
itself falls under one
fruit
of the
crops,
biodiversity
and in the
hotspots, and the
water,
area
we
where
are putting
farm
the water
is
plants, sir,
blessed
like
with
spinach,
bodies,
water spinach,
different
water
flora
mimosa,
and
that
fauna.
local plant is there.
The site of the
And
central
also
farm
fish
is
release.
situated along the Mericombe
You
Road,
see, sir, that
Lumfel
we have
Path,
developed complete.
Imphal East District. In recent years, under the able guidance of the Honorable Vice Chancellor, Dr. Anupam Mishra, the newly developed farm is made functional by a team meticulously led by Professor
And then
Indira
complete, we are
Saranyan,
doing also
Dean
5A
College of
approach. Means
Agriculture,
agriculture also
CAU
using,
Imphal,
agroforestry also using,
farm in charge,
apiculture also
Dr.
using,
S.M. Haldar,
then
Head Department of
fisheries,
Entomology,
all we are taking.
College of Agriculture, and estate in charge, Shri
Can
Chittaranjan
you
Singh, College of
produce the honey
Agriculture.
here?
Inaugural
Here
audit
to sir,
farm
no,
was done by
here not
Honorable
possible
Chief
because in
Minister of
summer
Manipur,
time we are
Shri
sending
N.
to,
Veer Singh, during the Agri Fair held
in winter we
January
can start, sir,
2021.
but summer time we can send a migratory type is
The
running
main
here.
components of the
Because that
CAU
area is
Central
totally,
Farm
sir, there
are
na
the
35
natural
temperature is
farming
there.
model
So we
unit,
are one place we can
integrated
throw out
farming
here hearing,
system unit,
but here not possible, sir.
agro-ecotourism site,
But we can make another
experimental
place in
research
Uttarakhand
area,
another that we can
protected
send, and
cultivation
October
area,
to March,
demonstration
tak,
fields,
no any problem
and a
here.
center of excellence for oil palm demonstration unit.
So we have also, sir, oil palm
The
project also there
50-hectare
given the
wetland
government of
area
India,
of the
and
central
it's
farm
21
was
crore
converted
rupees
into
project, sir.
farming land consisting of
And you
sunken
see, sir,
bed,
this we are
raised
doing
bed,
for
and
development
fish
of this.
ponds using tractors and JCB. The 5A approach of agriculture, agroforestry,
What do you want to
apiculture,
focus?
aquaculture, and animal husbandry for conversion of wetland
Yeah,
to
sir.
farming.
That's why I told, sir, this is related to our farm. If we are developing farm, then this we require, and on the basis of this we receive this award, sir.
Okay, very good. Congratulations.
Yeah, thank you, sir. And related to revenue generation, I completed 4 years in CAU, so I that generated 19.51 lakh from honey as well as boxes as well as pollen substitutes, sir.
And when you are going for IPM on the training and all, no, we can give training for this for producing honey nuts.
Yeah.
Beehives and other things. We can bring the people from tribals and other things.
Yeah, no problem, sir.
The tribals, SC, ST, tribals and all this, no?
Yeah, sir.
Here in and around are there, something like that, no? We can show them how to.
Sir, we have one group is there.
We can go there and show these things, some material you need.
Yeah.
So and you just think it over. We can give training on honeybee also.
Yes, sir. Yes. Actually, sir, we have already make one group, 500 group for farmers, and then we at present also we give the some technology and that.
ABI. In ABI, na? ABI
Yes, sir.
Yes, sir. Here. But somewhere in KVK something like that, training we can give.
Yes, sir. Yes.
Training we can give.
And we also generated sir revenue from IFS model also in central farm. At present around 15 to 20 lakh per year. Revenue is coming from this video that we are seeing 15 hectare area. And also we generate the revenue. Yeah. Yes, sir. I will try, sir. And then we also, sir, national conference we are conducted, and we also generated the revenue from this also. So this is about my publication, sir. This is related to some books we are published.
More than 10 factor.
Around 15 is there, sir.
You can apply for national.
Sir, I applied next year, and this year, sir, maybe 6-7 paper is coming my.
Okay,
so after that,
applied, are going to apply next year.
Going, sir, okay. Next year maybe. So this is about this publication. We publish, sir, entrepreneurship opportunity in agriculture. So we are taking from NBIR most of the 6-7 chapter we are taking live by control, trichoderma rearing, biopesticide rearing. This type we are putting in this book, sir. And we also publish this inventory. In CIU also we publish and another CIS also. So I request you, sir, our technology also there. So we make some inventory 30, 40 pages or 50 pages. So if anyone is coming VIP, we give, this is our technology. Inventory, that also we require, sir. Because we have completed 37 years, so some technology is there.
But if we are coming out with the technology development, that will be sufficient.
Yeah.
Why to inventory separate?
Actually, initially we can do certain inventory. After that, we can complete.
We are going to have technology developed and also success stories.
That we can do, sir.
And ultimately, we are going to proceedings will be easier. That they will be preparing it.
These are other publications, sir. Technical bulletin and other we have published. And this I received last year. I received this award, sir. 25,000 from that T.N. Ananta Krishna Foundation Award from Chennai, sir. And this another, sir, for stall. Any stall we are taking 20,000 for this also. And this also received from this Prawn Protection Association of India, sir. Then 15,000, sir. And Agriculture Today Award also there. I also request to our scientists, young scientists, can we apply for this also? It's very good given this award. Agriculture Today Award, sir. Young Scientist Award, only one given in India, sir. So these are the news and we are publishing this.
For you people, young scientists ordinance below 45 or 40?
40, sir.
40. How many of you are having less than age of less than 40? Dr. Meena?
Dr.
Case?